So gene expression, or some people call it protein synthesis. It is a two-step process. The first one is called transcription. Transcription is basically converting DNA into messenger RNA. We call this transcription. Location, it is in the nucleus. DNA serves as a template for messenger RNA formation. So it's just like what I just said, it's converting DNA into a messenger RNA. And now, second step is the translation. Translation takes place in the cytoplasm. So what does translation do? It converts the messenger RNA into a protein, or you can call it the polypeptide. Polypeptide is basically the primary structure of protein. Now let's take a look at this one. The sequence of the messenger RNA bases is complementary to the sequence of DNA. So this refers back to the transcription here. DNA serves as a template for mRNA formation. The way that messenger RNA can form is based on DNA base pairing sequence. So DNA, you know that it has ATGC, but RNA also has similar. It has A, but instead of having T, it will have a U. U is called uracil. And then it also has the same G, same C. So A, G, C, they're the same. But in RNA, instead of using T, thymine, it uses the uracil, U. And then in here, from the in here it said that the sequence of mRNA bases, that means A, U, G, C, it will determine the sequence of the amino acid in the polypeptide. So it's like, you may consider it's like in the spy movie. Okay, it is the original message, and then it is being transcribed into messenger RNA, and then messenger RNA, you have to translate messenger RNA into protein sequence. So it is like two levels of encryption. You may call it encryption. And the amino acid, as you know, there is the monomer to make a polymer protein. And polymer protein, it has a different level of structure. And the primary structure of the protein, we call it a polypeptide. So by translating the mRNA bases AUGC, we will get the sequence of amino acid to form the primary structure of protein. And in the process of translating mRNA into polypeptide, we require several helper. One is called tRNA. tRNA stands for transfer RNA. And we also need something called ribosome. At the beginning of the semester, we learned about cell structure and function. We learned about different organelles. One of them is the ribosome. Ribosome is made up of ribosomal RNA and also some protein combined together. So let's take a look at here. This here, from DNA to messenger RNA, from messenger RNA to polypeptide, we call this central dogma. So from DNA to RNA, from DNA to messenger RNA, we call it transcription. It takes place in the nucleus. transcription takes place in the nucleus, and the enzyme that it requires, it is called RNA polymerase. From messenger RNA to polypeptide, we call it translation. It occurs at the ribosome. But where can you find ribosome? You can find ribosome in two places. One, ribosome attached to ER, and we call it rough ER. Or you can find free floating ribosome in the cytoplasm. So, and what does it require to do translation? It requires, of course, ribosome. It requires tRNA. It requires ribosome. Location should be cytoplasm. Ribosome is part of the candidate, the helper for the translation. So location should be cytoplasm. You can call it rough ER or something like that. Then you have tRNA and ribosome as the helper for the translation. And then in here, you can see the structural depiction in here. So DNA, one is called coding strand, one is called a template strand. And then we use the coding strand to make the messenger RNA. So this is the coding strand. Sorry, we use a template strand. We use a template strand to make the messenger RNA. And the messenger RNA will consist of, since it is T, and you have the A here. The direction of making messenger RNA is the same as the DNA replication. Remember, in DNA replication, the newly made strand, or we call daughter strand, it is made from 5' end to 3' end. In here, the messenger RNA is also made from 5' end to 3' end here. So it is the same direction from 5' to 3', and it is complementary to the template strand. So because the DNA is T, then you get the A here, C, G, G, C here, C, G, T, A, so on and so forth. But if you get you get an A in the DNA, you get a U in the messenger RNA. So U is a uracil. We'll talk about that in the next few slides. And then every 3 bases, we call it a codon. What does it mean, a codon? It is every 3 bases, it will be translated to an amino acid. For example, this base here, AGC, it is translated into a serine, amino acid serine. For GAC, it is translated to aspartic acid. CCC, it is translated as proline. So every 3 bases, it becomes an amino acid. And then ribosome will join the amino acid, not the ribosome, actually there's an enzyme called peptidotransferase. Peptidotransferase is an enzyme that will connect the amino acid together by forming the covalent peptide bond. So let's start with transcription. So transcription, so a gene, what is a gene? A gene is basically a segment of DNA that can code for a protein. a gene will serve as template to make a messenger RNA molecule. These two are very, I would say, a very complicated definition. So you may just I would say, to make it simpler, you can just stick with the historical definition. A gene is basically a segment of DNA that can code for the sequence of amino acid in a protein. I think that is easier to understand. The proposed definition is basically the difference is that a gene can be either on the DNA or RNA, it doesn't matter. A functional product, meaning that the DNA has the sequence that can code for the messenger RNA, and the messenger RNA can code for a protein. So the new definition basically include RNA. Basically, they are more specific, or they include both DNA and RNA, and the product is both RNA and protein. But for the historical definition, is basically just go from DNA into protein. That's basically it. Messenger RNA. Messenger RNA is the product of transcription. So it serves as an intermediate between DNA and protein synthesis. Its form is the product of transcription, and then the process requires RNA polymerase. So again, ASC is a protein. It's a protein that makes RNA as a polymer. That means it is a protein that will elongate the polymer as a chain. So the RNA polymerase will bind to a promoter's region of a DNA. Promoter region is a special form, is a special sequence on the RNA in the DNA. It's a special sequence on the DNA that RNA polymerase can recognize and bind to it. After the RNA polymerase binds to this promoter sequence, promoter region on the DNA, then RNA polymerase will start to do its work to replicate. To transcribe DNA into RNA. So the DNA helix, it is a double helix. It will also unwind, but this time it is also done by RNA polymerase. And then RNA polymerase will unwind the DNA helix, and it will also grab the RNA nucleotides in the nucleoplasm. Nucleoplasm is the fluid filled inside the nucleus. So RNA polymerase will grab the RNA nucleotides in the nucleoplasm, and then it will connect the nucleotide complementary to the DNA sequence. And in this case, a chain of messenger RNA can be formed. So whenever you form a chain of nucleotide, it has to be complementary to the original strand. In this case, as I mentioned before, in DNA we have ATGC, but on RNA we have U go with A still go with T, C still go with G, G still go with C. So the complementary base pairing rule between DNA and RNA, most of them are the same, but if in the DNA you have A, then RNA you must get a G, a U, uracil. So this is how a transcription occurs. First of all, the RNA polymerase binds to the promoter region on the DNA, and then after this, you will start to synthesize the messenger RNA molecule, which is purple in color. And again, just like DNA replication, the messenger RNA is start with the 5' end and then end with the 3' end. So it is also from 5' to 3'. And then as the RNA polymerase go along this, we call it template strand, then it will transcribe the bases from the template strand into RNA bases. The RNA sequence, as you can see that if the DNA has the A, the messenger RNA will have the U here.